pathway. So let's now talk about this pathway. You already know, I have already told you the pathway. So it converts like this. So you know this glucose 6-phosphate converts in glycolysis. This glucose 6-phosphate in glycolysis converts into this fructose 6-phosphate through one step process, and this fructose 6-phosphate will. So this is a divergent pathway. That is why this is called shunt, and this has two stages of pathways. Simple is oxidative phase. One is oxidative. This is irreversible oxidative, because oxidative, you know, carbon dioxide, one carbon dioxide is removed here. So this is irreversible, and this is reversible state. Non-oxidative is a reversible state. This is irreversible, this is reversible. Means, so from glucose 6-phosphate, it is irreversibly converted into pentose 5-phosphate. But if a cell requires only ribose 5-phosphate, this fructose 6-phosphate can convert into this. Okay, fructose 6-phosphate can convert into ribose 5-phosphate. Fructose 6-phosphate can convert into directly into ribose phosphate. It does not need to go through this oxidative phase. So this is the difference. You can see if a cell requires this only ribose 5-phosphate, then the pathway will be, it can be easily synthesized from fructose 6-phosphate. There is no need for synthesis of this. For example, protein producing cell, the cell which only produce, it is not dividing, only the cells which are secretory cells, for example glands which produce insulin. So in that case, what RNA has to be produced, okay, and that RNA require this. So this fructose 6-phosphate can easily be converted into a ribose, okay, ribulose to ribose. There is no problem. But if some cell requires this and NADPH both, then it has to go through this. Or any cell, if it requires NADPH only, in that case also it has to go through this pathway. So oxidative phase is for production of NADPH, and for production of ribulose, oxidative phase is not required. Okay, this is the very important point. So oxidative is NADPH. Now you can see this enzymes. Enzymes, you can see two. Just memorize this two enzyme here, two enzyme here. That's it. So two enzyme is G6PD. This is more important enzyme. Here also this is more important enzyme. G6PD, because G6PD deficiency causes hemolytic anemia. Now here phosphogluconate dehydrogenase. Second, both are dehydrogenase, and this both dehydrogenase will produce two NADH, one NADH from here, this, and one NADPH from this, not NADH, NADPH from this. Here you can see transcatolase and transaldolase, both are transferase. These are both are oxidoreductase, these both are transferases. Transcatolase, you can remember transcatolase, you can remember two from this. So it transfer 2 carbon. So that is how you can remember it, transfer 2 carbon. And aldolase transfers 3 carbon. So you can differentiate between catolase and aldolase. And this, see, there is one rule, aldose sugar is always acceptor. It is always acceptor. And keto is always donor. Okay? Keto is always donor, aldose is always acceptor. If you remember this, you will remember the pathway, everything about pathway. But you don't have to remember the whole pathway. If you remember this much, and that is sufficient for your question, solving your question. And do you find this pathway difficult?
No, sir.
No, it's very easy. 2-phase, this is a shunt pathway. Shunt pathway, this is a part of glycolysis, you can see. 6-phosphate. And this is shunt pathway. 2 stage. If you see steps, there are many steps you can see. 3 steps in irreversible, 3 steps here. But 2 stages is sufficient for your memory. Okay, and this NADPH. Remember, NADPH means cellular reductive biosynthesis. We have already done reductive biosynthesis of, we have done fatty acid. Reductive biosynthesis of fatty acid. We have done reductive biosynthesis of, we have done cholesterol. Reductive biosynthesis of deoxynucleotide. So these are for this NADPH is required, reductive biosynthesis. And detoxification, cellular detoxification. It can be detoxification of free radical in RBC, or it can be detoxification of xenobiotics, foreign substance, foreign toxic or non-toxic substances. Xenobiotics. Xenobiotics, foreign is drug that you take. So these two detoxification require NADPH. So remember cellular reductive cellular biosynthesis and cellular detoxification. These are very, very important function of NADPH. If you remember these two terminology, you can actually tag everything with this terminology. Now, transketolase becomes important because in condition of vitamin, because it is dependent on vitamin B1, what other enzymes that are, can you name some enzymes that have vitamin B1 in addition to transketolase? Other enzymes that have vitamin B1? Yes? Can anyone? Try to be quick.
Alpha ketoglutarate.
Very, very good. Very, very good. Very, very good. Thank you. Alpha ketoglutarate dehydrogenase. And then also pyruvate dehydrogenase. And then third is which enzyme? Branch chain CAD, BC-CAD. Branch chain keto acid dehydrogenase. This is in, we will study this in leucine, isoleucine, and valine catabolism. So these three have similar structure. It all have a vitamin B1 to B5, vitamin B5. So I told you to remember B4 as lipoic acid. So that is how you can see B1 to through B5. It all these contents. So if you see only these three vitamins, these three enzymes, and this transketolase has vitamin B1. So in that sense, I'm also teaching you vitamin B1 function. So transketolase actually activity test is done in laboratory to find out the vitamin B1 availability. Transketolase activity test. Transketolase activity test is a test that is done to assess B1, vitamin B1 availability or deficiency, vitamin B1 deficiency. Okay, if activity is good, then result will be normal and vitamin deficiency is not there. If low activity, vitamin B1 deficiency, you can find out. So this is the significance of clinical significance of transketolase. Transketolase test is done to assess vitamin B1 deficiency. Very important. Okay, here this is associated with G6PD deficiency is associated with hemolytic anemia. These two are very, very important clinical correlation with this pathway. And you can easily understand ribose 5-phosphate will convert into PRPP. We have already done this PRPP. It converts into PRPP synthesis, PRPP, and then PRPP is required for synthesis of everything, DNA, RNA, other nucleotide, even this NADPH, including this NADPH. In NADPH, NAD, FAD, FAD. So these all are synthesized because of this ribose 5-phosphate. Now, so you already know this.